BTEC Applied Science for AAQ, Unit 1 Biology, B4 we've got up to, and this is an introduction to DNA. So what is DNA? It's deoxyribonucleic acid is what it is. What does it do? Well, it's basically recipes for making different proteins. Yeah, lots and lots of recipes for making lots and lots of different proteins. How does it do that? Well, now we know that there's about 20 different types of amino acid, and these amino acids join together in different combinations to make proteins, and there's millions of different kinds of proteins. And then the proteins will combine to form a living thing, and then all of the stuff that the living thing needs to stay alive. Now, the DNA tells the amino acids, in other words, which amino acids and in what order to join together to make particular proteins. Yeah, bits of the DNA correspond to different proteins. And then if you get it all right, then you end up with a particular organism, like Kevin the kangaroo. Okay, so DNA tells the amino acids how to join together to make the proteins needed to make Kevin the kangaroo. Now, the actual structure of DNA, talk more about this later. It's like, imagine it's like a ladder. Now, the ladder is actually twisted into a double helix, but forget that for the moment. It's like a ladder. So you've got the rails of the ladder, and then you've got the rungs of the ladder in between. Now, bits of the ladder are made up of units called nucleotides, and all of these nucleotides looks a little bit like ATP, doesn't it? Yeah, and there is a bit of deoxyribose in there, isn't there? That's the sugar there. So you've got sugar phosphate backbones. Yeah, that's the rails of the ladder. And then in between the rungs of the ladder are base pairs. Yeah, pairs of bases. There are 4 bases in DNA, which are cytosine, guanine, adenine, and thymine. Now, T pairs with A, and A pairs with T, and G joins with C, and C joins with G. Okay, so basically it's the A's and the T's get together, and the G's and the C's get together. And they join together with hydrogen bonds. You'll notice that the A's and the T's are 2 hydrogen bonds, and the G's and the C's are 3 hydrogen bonds. Okay, so that's the basic structure of DNA. We've got this sugar phosphate backbone, in fact 2 of them, and they're made up of nucleotides. They're the base units that make it up. And then the 2 bits of ladder are joined together by these base pairs, and there are 4 bases that in DNA. Now, almost every, spelt wrong, cell in our body contains about 1.8 meters of DNA. In fact, nearly every cell in our body has exactly the same DNA. And if you stretched it all out into a line, it would be about 1.8 meters in a tiny, tiny, tiny little cell. And a single strand of DNA contains millions and millions of these letters, of these base pairs. Now, normally it's just like, not completely random, but it's uncoiled in the nucleus. It's only just before cell division that it organizes itself into these chromosomes ready to split. Yes, just before cell division, the DNA organizes itself into chromosomes. In fact, that's the only time you can actually see the DNA. Before that, it's just too spread out. Now, the DNA is in the nucleus, and what happens is that parts of the DNA, say that it's to make a particular protein, then part of the DNA is copied, and the copy of the DNA is called RNA, and that process is called transcription. You imagine there used to be people called scribes, and their job was to like copy out bits of the Bible or whatever. Yeah, scribes. Transcription is to copy bits of DNA. And it's special chemicals do this. It's called RNA polymerase in the nucleus, and they make copies of parts of the DNA. And what you end up with is this stuff called messenger RNA or mRNA. And that can leave the nucleus because it's small and it can fit through pores in the nucleus and go into the cytoplasm. Notice that it's just like one side of the ladder. Yeah, half of the ladder is missing, and that's RNA. So DNA is the whole double helix thing. RNA is just one half of it, and it's just a small section of it as well. Now, translation, what happens is that this piece of RNA, messenger RNA, contains the information to make a particular protein. And there are these things inside our cells, these organelles, which are called ribosomes. And ribosomes are factories for making proteins. And what happens is that the bit of RNA, messenger RNA, goes in one end and it chugs its way through, and then 3 letters on the RNA, yes, is the code for a particular protein. So they read the RNA code 3 letters at a time, and a particular amino acid which is floating around gets sucked in and it gets tagged on to the end of the protein. And it keeps doing that again and again and again until you've actually made the protein. Yes, and as the protein forms, it coils up and it forms a particular shape. We know that the shape of proteins is very, very important for them to do their job properly. And the 3 letters in the code tell the ribosome which amino acid should be grabbed from outside and added to the protein next. So ribosomes are the factories which make proteins, and they get the information to do that from the messenger RNA. And then if you've got all the right proteins in the right place at the right time, you've got Kevin the kangaroo. When I was doing my refreshing up on this particular topic, because I was a bit rusty, I came across some very, very good YouTube videos, which I do recommend you have a look at. So if you want to pause the video and have a look at these, and they explain what's going on very, very well.